I'll tell you today about what we are doing in terms of trying to understand how metastasis are working, and also definitely how priming, and I will, yes, Clotilde talk about EVs, of course, but I will introduce other stuff because I think it's connected and it's important to mention it. So I don't think we've heard already many talks in this meeting about the importance of understanding metastasis. It's still a major issue, health-related issue, as 90% of the patients, that it's going to lead to the death of 90% of the patients when they have metastatic cancer. So mortality of metastatic cancer at this stage is still stagnating or even progressing for some cancer types, and there are strong, very urgent need to develop new treatment strategies. And we thought that at least when I started my lab, that we still need to work and have additional efforts on trying to understand the mechanism and also the mechanics, and you will hear about that today, of this phenomenon, to understand it better and then develop new treatment strategies. So in the past, we've worked on a few aspects leading to tumor metastasis, but concentrating our efforts on understanding primary tumor development and invasion features. And we've done some work where we could show, actually, and this is something that has been shown and visualized by many labs over the world, that the tumor-associated stroma is something that is essential in shaping invasive tumors, making them very metastatic, by increasing both the stiffness and the anisotropy of the extracellular matrix that surrounds them. And this leads to increased metastasis. But there are, of course, definitely additional features that contribute to that. And the communication that occurs via extracellular vesicles, as you can see in this tiny movie here, definitely is something that will shape the invasive and the metastatic behavior of tumor cells. And we need to understand this a little bit better. So I'm using these two different approaches to introduce the fact that at some point I thought that we would need to have an improved resolution of the imaging techniques that we are using to uncover additional insight into metastasis. Oops, and this is not working. I'll launch it again. So we decided a few years back to launch and develop a new technique which we call Intravital correlative light and electron microscopy, which is basically, I won't spend too much time on it, but the overall idea is to combine the power of intravital imaging that you can perform in living animals. And this is one example where we image one tiny tumor xenografts in the mouse ear that can be imaged and dynamically recorded over time. And then try to come up with correlative methods that allow you to focus on one single region of interest once the animal is sacrificed, that you can then fully characterize using a volume electron microscopy. And this is basically what you see in this movie here, which is the same region of interest that has been visualized using intravital imaging, but this time fully mapped using serial transmission electron microscopy. And the end of the movie shows you even electron tomography, which shows you how we can very strongly increase the resolution of the imaging that we can provide to specific region of interest within cells of interest, and can definitely show that you can have vesicular resolution, which is essential, for example, if you want to apply this to EV biology. But then we decided to switch a little bit gears. As I told you, we focused for some time on primary tumor, but we then decided to look at more late stages of metastasis. And what this scheme represents is actually the vascular beds that are pro-metastatic, and that would be, of course, subjected to priming and to the entry of circulating tumor cells favoring metastatic progression. And we asked a few questions. So what happens at distant sites from an hemodynamic point of view? What is the impact of fluid mechanics? And it's an essential feature when you are dealing with extracellular vesicles. How do cells extravasate? And I'll show you one example of that. And of course, how does EV-mediated priming occur in vivo and at real time? Because this is our main motto, is to really look within the animal and at real time at these events we're interested in. And I'll show you today three different aspects of these studies, how we think that the intravascular arrest is occurring in these vascular beds, how we could actually identify a new mechanism for the extravasation of circulating tumor cells, how we develop technologies that allows us, and hopefully you in the future, to track the priming of these metastatic niches by extracellular vesicles. So I'll start very quickly by just mentioning, because it's all linked also to the biology of extracellular vesicles, but focused on circulating tumor cells. We spent quite some time at developing methods using an experimental metastasis model in the zebrafish embryo, which we are using here as a highly sophisticated microfluidic system, because as you can see from the movie, we can have access to blood flow circulation very easily, quantify it, but also tune it. This is what you see in the tiny scheme here. By using drugs that would alter the pacemaker activity of the heart, we can change flow profiles very easily in this embryo. We've developed it to use it as an experimental metastasis model, so we IV inject circulating tumor cells, and this is just an example that happens at individual tumor cells once they're injected, and you see a few examples of cells that are still intravascular, submitted to the shear forces of the blood flow and also the collision of the red blood cells. The constriction forces that this cell is also facing when it's trying to move within the vessel at another site to actually meet another tumor cell, and how these cells are capable, because this is a bunch of cells that have extravasated, and you see another one that is in the process of extravasated at this stage. I'm just using this movie to highlight what happens in this region of interest and how this is related to the flow forces. Because when we looked at a high number of embryos at the position of arrest of circulating tumor cells, as said, in high numbers, we could highlight a hotspot of arrest that actually fits very nicely with a region of hemodynamics that correlates with a velocity drop. So basically, the circulating tumor cells would arrest in regions where the hemodynamic profiles are favorable for their arrest. And it was actually very interesting to notice that similar studies had been done in human patients bearing brain metastasis, where they did a correlation and a mapping of the position of macrometastasis in a cohort of 100 patients having brain metastasis, to which they correlated the blood perfusion profile, and it turns out that it matched exactly what we've observed in the zebrafish embryo, which is basically that blood flow perfusion is actually has a very strong impact in the position where circulating tumor cell will arrest and will eventually form metastasis. Second step, how do cells extravasate? This is also when I started my team, and even before that, something where I saw that it remains a big black box in the field. And we thought that with the development of intravital correlative light and electron microscopy, we could provide additional insights. So we had to redevelop the technology, because we now are capable of applying this technology to single cells that are in the process of metastasis formation in a living mouse brain. So what you see is an example of a single tumor cell here in green that is still intravascular at that stage, and that is in the process of performing extravasation in a living mouse brain. And we've developed, I won't go into details of that, but again, additional technologies that allow us to remap and fully map this same tumor cell that you see here from the intravital imaging point of view using electron microscopy. And we're using additional methods such as X-ray scanning that we perform on the EM embedded sample. But I won't go into details of that. I want to go directly to what we are capable of obtaining. So we can correlate, again, the intravital imaging of single tumor cells imaged in a living mouse brain to nanoscale imaging of the same tumor cell using volume electron microscopy. So the movie that you're looking at is a FIB-SAM movie, so scanning electron, focused ion beam scanning electron microscopy, which provides an isotropic resolution of 8 nanometers. This movie has 7,000 frames, and it fully maps the cell that is still intravascular at that stage. And the movie stops at a region of interest where you would see still a tiny piece of the cytoplasm of the tumor cell, poking a tiny figure between two endothelial cells, and you would see here the basal lamina of this brain capillary. So we use this technology a lot to try to understand how these cells would eventually extravasate. And I'll show you 3 examples, as we think that ICLAM is capable of really unlocking the mysteries of metastasis. This is an example of cells that had just extravasated. So we are a few hours after the extravasation of this blood vessel. You can see a bunch of 3 tumor cells, and basically the type of connections that they make one to another. And an example, and this is something we're now dissecting a little bit more, is, for example, looking at organelle positioning in these very early steps of extravasation and macrometastatic outgrowth. This is an example, as I'm pointing it here, it's actually the only example on these 50 cells that we fully mapped where we could actually catch exactly a cell that is in the process of extravasation. So as you can see from this volume electron microscopy reconstruction, that you can still see one region of the cell that is intravascular, while there's another region of cell that is fully intravascular, showing this classical example of diapedesis through active transmigration through the vessel wall that everybody can see in the textbook. But it was the only example that we could, although the imaging was dedicated to really catch this event very, very accurately. So we thought for a long time what would be the explanation, and we came up with ideas by looking very carefully at cells that are still intravascular, so very close to perform extravasation. And when you look at these cells, and this is just an example of different Z steps that I'm showing you with the segmentation to really highlight the endothelial cells, so the cells that make up these capillaries, is that when you look very carefully at endothelial cells that are very close to arrested circulating tumor cells here in green, you can see that they're very quickly sending tiny filopodial and extension protrusions very close to the disposition of arrest, with even the potential of creating additional lumen. And this is an essential feature of the extravasation method, because this event is actually massive, and most of the tumor cells would actually trigger these endothelial protrusions very rapidly after the arrest, again creating new neolumens, like strong projections, and even blocking the lumen of this tiny capillary. And I'm not showing you the full result of that, because the paper has just been published recently, providing all the electron microscopy images, but we have now reasonable evidence to show that there is a very strong intravascular remodeling that is actually one of the key features, at least in the context of brain metastasis, that triggers the extravasation of these tumor cells and then subsequent metastatic outgrowth. Then, of course, the next question is how do flowing extracellular vesicles prime metastatic niches, and this is the main focus of the meeting here. And we have a habit of saying that Clotilde Thierry, and I'm not saying it because it's the chair today is our EV queen, but I have my EV king, which is Vincent Yen, as Clotilde mentioned. He's the person that is doing most of the EV work that we are doing in the lab. And with Vincent, we're asking many different questions that I listed here, and I will go through some of them. But basically, the main idea is to come up with imaging methods and of course, molecular approaches to try to understand how this priming is occurring live at real time in vivo within the animal. And again, we used the zebrafish embryo as a first model system to understand that from an imaging point of view. This is one example of the caudal plexus of a zebrafish embryo, where we can see endothelial cells in white, macrophages in green, and some extracellular vesicles that are here labeled in purple that were IV injected within this animal. This is a few minutes after the injection, and you can see that it's highly dynamic and that macrophages, and I will come to that, engulf a huge amount of these extracellular vesicles. But I'm showing this movie to highlight the fact that the caudal plexus in the zebrafish embryo and in our experimental metastasis model is the vascular hotspot for metastasis. And it's actually very highly amenable to confocal imaging at high resolution and at high speed, so which makes the whole study way more easy. Why? And that was one of the first things we had to confirm, as it had been shown by many groups over the world, that the EV in the context of metastasis has this very strong priming effect. Does it have it in the zebrafish embryo? And yes, this is just a quick example to show you that when we compare the priming that occurs with beads, and we have now methods to show that with non-tumoral EVs it would be similar, in comparison to animals that have been primed with extracellular vesicles, and this is the timing of priming that we've imposed to this embryo, we can massively potentiate the metastatic outgrowth within the zebrafish embryo. So it's a very relevant, highly relevant model to look very carefully at how this priming is actually occurring within the living animal. And it turns out that here EV are now labeled in cyan. When you look very carefully at this caudal plexus region, so this is another example where we used transgenic lines that has red blood cells labeled in red, endothelial cells in green, and we've injected cyan extracellular vesicles. Looking at them again a few minutes after the injection, you can see that we can very nicely track them within this living zebrafish embryo. So can we actually track them from at real time? This was one of the first examples we've been able to provide, when looking at the dissemination of injected extracellular vesicles within the blood circulation of this embryo within the caudal plexus region. And you can see that it's, of course, highly dynamic, subjected to blood flow effects. And we had, of course, to adapt the imaging technology. So what you're looking at is a movie recorded at a frame rate of 100 frames per second, so that we could actually fully map the dissemination of these extracellular vesicles. And you can notice already from this movie that very quickly you see clusters of extracellular vesicles that happen a few minutes after the injection. You see events of arrest that occur very quickly. So it's a very cool model and a very interesting region of interest to go a bit further. By using another transgenic line that allows us to track individual macrophages, we've quickly realized that the clusters that are formed within blood vessels, so I really insist on the fact that what you're looking at here is within blood vessels, corresponds to a subpopulations of patrolling monocytes that have this potential of and this property of engulfing huge amounts of extracellular vesicles that have just been ejected. Because if you compare it to this macrophage, which is fully extravascular at this stage, you can see that the difference in internalization, of course, is very obvious because this one is fully extravascular. So it was actually very interesting to look a little bit further in the biology and the behavior of these patrolling macrophages, because they are highly dynamic cells. They really scan the surrounding. They are stably anchored to the vascular wall, and they have this property, of course, of scanning and engulfing a huge amount of extracellular vesicles. Having developed CLAM in our lab, it was definitely a good example to use CLAM and come up with additional insights on where these extracellular vesicles actually would end up once they have been engulfed by these patrolling macrophages. So this is another example where we could look at two patrolling macrophages within the blood vessel. You can still see that this blood vessel is still flowing EVs, a huge amount of extracellular vesicles internalized, and we scan this region using correlative light and electron microscopy. So this is the reconstruction of the two cells by confocal imaging and what we can obtain by volume electron microscopy once we've relocated the cells. I pass the details of the technique. It's quite time consuming, but once you have fully mapped the region of interest, it's quite insightful. So we've looked a little bit at these patrolling macrophages, and because I think that they have not been studied enough in the field, and we should be looking at these subpopulations a bit more in depth. And these are just a few electron microscopy images examples of how these cells are actually stably anchored to the vascular wall, which is something that we would be interested in the future, and how they use this very ample membrane sheet to actually be able to scan the surrounding and engulf whatever is around them, including extracellular vesicles. So when we use correlative light and electron microscopy a bit more now, in moving into the resolution and try to correlate the position of the internalized extracellular vesicles from a confocal point of view, which is the first line that you see here, to the position from an electron microscopy point of view. So this is correlating basically the signal that we obtain from the confocal imaging to where we think it could be correlating to in the patrolling macrophages. It turned out that most of the signal was actually correlating to late endosomal and lysosomal compartments, suggesting that these extracellular vesicles are actually potentially very quickly degraded by this patrolling monocyte. These are just additional examples that you can find in the paper we published back in 2019 of additional examples of extracellular vesicles. We cannot be correlative at this stage, so we have no way to say that these extracellular vesicles are actually extracellular vesicles that we have injected, because the correlation is not as precise, but it definitely highlights a few of the strategies that are used by extracellular vesicles to be internalized. But when you look a bit more carefully at this movie, so this was one example, strong internalization by patrolling monocyte, but you, of course, see that the endothelial wall is actually quite efficient also at engulfing a huge amount of these extracellular vesicles. So we spent quite some time trying to understand how the internalization from the endothelial cells point of view is actually occurring, with again the idea of trying to understand whether flow and fluid mechanics impacts this, and what allows the arrest of these extracellular vesicles at the surface of the endothelial wall. And I will very quickly go through that. We started by doing a few sets of tracking of red blood cells and extracellular vesicles behavior in single blood vessels. So this is the tracks that you would obtain when you look from a high-speed imaging point of view how they behave. And when you compare red blood cells to basically the dissemination of extravesicle within the blood vessel, you can see that the behavior is quite different. And thanks to the hemodynamic Poisson law, these extravesicles actually are very quickly put in contact with the vascular wall, where they would engage in potentially a rolling phase. This is something that we cannot assure 100%, but we can definitely see a slowdown of the extravesicle at the surface of the vascular wall, suggesting that there is active adhesion that occurs when they arrest within this region of interest. And indeed, we had some additional insights that the enrichment of cell adhesion molecules at the surface of extravesicles would be one of the triggers. And this came from other studies that we performed in the lab, and that also come from the expertise of Vincent in the group, where he had identified in the past that RAL1 is actually an essential GTPase that leads to the biogenesis of multivesicular bodies in C. elegans. And we applied this to our tumor models, which is in that case the 41. And when we knock down the RAL-A isoform and the RAL-B, you can notice that the number of multivesicular bodies that are within cells is strongly decreased, again suggesting that it's really involved in the biogenesis of multivesicular body. And this leads, I'm not showing you the results here, to a very strong decrease in the number of extravesicles that are secreted, but it also affects the content of these extravesicles. And at the same time, knocking down these two GTPases leads to a very strong reduction in our triple-negative breast cancer model. So again, 41 that we're using mostly with the biopsy background mouse models. So if I go back to the content that is affected by RAL-A and RAL-B on their way to process extravesicles, we noticed that one molecule, CD146, was one of the key features from this comparative proteomics that we applied to the EV populations coming from these different cell lines. It turns out that CD146, so it caught our attention because it's a cell adhesion molecule that is classically expressed at the surface of the endothelial wall. But this time we've been able to locate it at the surface of extravesicles. So our idea then was to try to see and understand whether inhibiting the adhesive potential of this molecule at the surface of extravesicles would have an effect on the priming of extracellular vesicles. And I'm not showing you the full story, but just this one experiment where we used the famous priming model by having this IV injection model, 2 steps of injections of extracellular vesicles. But in one of the conditions, we've actually co-incubated the extracellular vesicles with an antibody that blocks CD146. So, well, I don't need to spend too much time on that. When PBS is injected, we have lung metastasis model in this tail vein model using the 41 system. We potentiate lung metastasis when we inject EVs from the 41 model, but when we co-incubate these EVs with an anti-CD146, we reduce strongly the priming potential of these extracellular vesicles. And in our minds, this strongly suggests that CD146, because we have additional evidence that it decreases the ability that these extracellular vesicles have to engage with the vascular wall and then be internalized in endothelial cells, is a strong feature of the way extracellular vesicles are actually priming these metastatic niches at distance. But then the other part of the story is we've noticed very quickly, and I'm just putting this movie and this additional image here, that there is a very strong correlation between the position of arrest and internalization by endothelial cells of extracellular vesicles and the flow profiles. Because if you compare this vessel here, which is the dorsal aorta, the main artery of the zebrafish embryo, to these tiny vessels here, and even this caudal vessel, which is called the caudal vein, you can see that the amount of extracellular vesicles that we can locate is quite different, suggesting that flow profiles control very actively the arrest and potentially the internalization of the extracellular vesicles. So we've used again CLAM just to make sure that these extracellular vesicles we're looking at from a confocal and a live imaging point of view are actually located within endothelial cells, and they are. Unfortunately, we don't have, with our CLAM technology, the resolutive potential to say to which compartments they're actually corresponding in these endothelial cells, because the endothelial cells are very thin cells that even from an electron microscopy point of view, it's very tricky to make sure that this fluorescent signal would actually correlate to a specific subcellular compartment. But we've decided to move on, and we started to try to really make sure that there is a strong correlation between the flow profiles and the ability that the extracellular vesicles have to be engage with the vascular wall. So we used flow mapping that is based again on high-speed imaging of blood flow profiles that you can do very easily in the zebrafish embryo, as you can see here from this movie, and correlated to the number of EV that are actually located within the endothelial cells. And there's an inverse correlation, so the lower the flow profiles, the better the EV uptake in this position. But it's not necessarily the lower, it's actually, it turns out that there is a very permissive flow profile that would favor this uptake within endothelial cells. We can tune this, again, as I told you, we can tune very easily the pacemaker activity of the zebrafish embryo, which in that case, using lidocaine, reduces the flow profile. And when we do that, we strongly reduce the amount of excess vesicles that are internalized in endothelial cells. Again, suggesting that there is a tiny range of flow forces that would favor the internalization. And we then combine this with microfluidic system that allows us to be even more precise in the way we control flow profiles, using an endothelial cell monolayer and looking at EV uptake with that case in conditions where we have primed endothelial cells or not with flow. And again, what this shows you is that the flow is actually very good in priming, in having increasing the uptake, suggesting that there's, again, as I told you, a permissive flow profile that would favor the uptake. So we looked at it a bit more in that. Okay. So these actually turned out to correspond to LAMP1 positive compartments, which turns out to be non-degradative. And I will very quickly go through that, because when we looked carefully, what you're looking at here are LAMP1 positive compartments co-localized with extracervicals in the no-flow conditions and in the flow conditions. And this is actually a magic red image. So magic red allows you to label cathepsin B positive, so degradative lysosome. And it turns out that the extracervicals have the ability to escape these degradative compartments. We've then used a tool that came from the lab of Alisa Weaver, which allows us to use the pH sensitivity of pH fluorine to basically have an indication of what is the pH of the compartments that are used by the extracervical. And it turns out, and I will go unfortunately quickly on that part, that flow is capable of rerouting extracervicals towards more neutral compartments. So basically, it's potentially escaping degradation. I'll go quickly through that, but we think that we've identified which compartments are these, and it turned out that there are RAP14 positive compartments, which had been shown recently to be non-degradative and non-acidic compartments. And just to finish, does this have a potential effect? So we've used transcriptomic analysis of endothelial cells that were subjected to EV and flow at the same time, and comparing the different conditions, we've been able to show that actually we knew that EV have the potential of priming angiogenesis in endothelial cells, but it turns out that flow is an important aspect in allowing this effect to occur, and this has been confirmed now in the zebrafish embryo system, where we have additional evidences that show that the conjunction of flow forces that allow the internalization of extracervicals and, of course, uptake of extracervicals is capable of tuning the pro-angiogenic potential of these endothelial cells. So this is a summary of this part, which is not fully published at the moment. And I'll just end up by doing a very tiny poster alert, because we have one PhD student here that will show his poster today at 4 o'clock, where we are trying to understand a bit more how we could potentially, so it's very different subjects, but how we could potentially prime anti-tumor response with a pox virus-induced extracellular vesicles, because Luca is actually working in a biotech called Transgene, very close to our lab, and we've teamed up to come up with ways to show that yes, extracellular vesicles are actually stimulated, their secretion stimulated upon viral infection, and that when we use these extracervicals, we have potentially a way to prime the immune system in a specific model, but he will tell you a bit more on that part. So just as a take-home message, what I showed you, and I'll focus on the last parts, we've been able to develop methods to track extracervicals in vivo at real time, to show that the cell adhesion molecule potential of extracervicals is essential in allowing their arrest and their internalization within endothelial cells, that blood flow actually controls very actively the uptake and the fate of these pro-metastatic extracervicals, and that endothelial cells, which are of course flow sensitive, are capable of rerouting extracervicals towards non-degradative compartments, and we think that this is an essential feature of allowing extracellular to have their job. So the work has been done for the EV part mostly by people that are highlighted here with the stars. And this is our new building in Strasbourg, and I hope I'll be able to see some of you in the soon future there. Thanks a lot.
Thank you very much, Jacky, for this beautiful presentation, and that highlights how important it is to do in vivo studies. We don't have much time for questions, so we may have like maybe two burning questions. So,
thank you for this huge amount of imaging you showed. But you say there is this arrest of EVs, if I understood correctly, on tumor cells, not on on endothelial cells, or?
On endothelial cells.
It's on endothelial cells, and then there was one image where you showed that there is a certain role of lidocaine, like this local anesthesia. Is it somehow related to the pH value, or do you can you explain the role of this local anesthesia? Is this increasing the arrest of the EVs, and what would this mean?
So we're using lidocaine as a sodium channel blocker, decreases the pacemaker activity of the heart, and this allows us to tune the flow profiles. We have additional methods to do that, and of course, and yes, indeed, it decreases the ability that the extracellular have to engage with the endothelial cells. So flow forces basically impact the ability that the extracellular would have to engage with endothelial cells and be internalized in that particular condition. And we have the opposite behavior when we increase the pacemaker activity of the zebrafish embryo. We increase flow profiles, and we have similar observation. Yeah. Thank you.
Thank you.
Beautiful talk, Jacky. Provocative question for you. Given the limited concentration of tumor-derived EVs in circulation and a fully mature immune system like in humans, what's your take on the role of tumor-derived EVs in metastasis priming?
Well, it's a very open question that we would love to be engaging in at some point. So at least the first aspect of your question relates definitely to a question we are interested in, is what is basically the amount of extracellular that are progressively released by tumors over time. So we are brainstorming on methods to be able to do that. At this moment, we don't have this methodology. I don't think anybody, even in this audience, have ways to know how many EVs are secreted and what's basically the frequency and the amount over time. So I cannot tell you. So we have to use this. They are not acute, I would say, because we're not injecting massive amounts. But these methods to have a first insight on how this is actually working. In terms of the fully mature immune system, this is fully open. Of course, we're doing additional studies trying to understand how the immune system, as many of you are doing, is primed by EVs in mouse models that are more adapted and, of course, fully mature in that case, so using syngenic system. And we have some insights at the moment. Yeah.
Thank you.
Thanks, Jacky. So I'm just wondering, did you guys deplete the macrophages to see whether there's a phenotype?
Yes, we've done that in the zebrafish embryo. Unfortunately, we did not have that much of an effect in terms of priming or like promoting or inhibiting metastatic outgrowth. So we're still struggling a little bit with the methods. The depletion works very well. What I can tell you is that we've done additional efforts on that particular aspect because, of course, we're definitely interested in the potential contribution of these patrolling macrophages to metastatic outgrowth, whether it's inhibiting or promoting it. And we've done some transcriptomic analysis now that suggests that potentially they could be linked to favor the recruitment of neutrophils, for which we have additional insights for our mouse model studies. So this is something we're trying now to make converge in the lab, definitely.
So thank you. I think we are a bit late and we still have the featured abstract, so I urge you to come talk to Jacky next. Jacky, you will be here for the rest of the
Sure, yeah.
Great. Thank you very much.
Thank you.